Hi, everyone. Thanks for joining us for today's webinar. This is the last one in our 2020 Vineyard webinar series. So hopefully everyone has enjoyed being on these webinars, and we can do something like this again next season. So this last one will be about what to do from this point, which is veraison, into harvest over the next couple of months. It's a very exciting season for all of us, as we know. So I'm glad that we have a chance to kind of regroup before this all starts. Today, Amaya Atucha and Christelle Guedot will both be speaking, as well as me. So first up is Amaya. She'll be talking about harvest targets for cold hardy varieties and how to evaluate ripeness. And then I will be talking for a few minutes about bird control options and what the research says about how those compare in effectiveness. And then Christelle will be talking about harvest insects until 1:45 or so. So we'll have 10 or 15 minutes at the end for questions. Just as a reminder, we've covered a number of topics in previous webinars this season. You can see those topics here. The most recent one we did was just last week on sour rot, botrytis, and bunch stem necrosis. So you can find all of these on the U of M Extension Small Farms channel. And I know you can also find them on the UW Fruit channel. Christelle or Amaya, how do they find that info?
I guess it would be the UW Fruit Program YouTube or something like that.
Okay, great. So you can go to either of those places and find the recordings of our previous webinars. All right, so I'm going to stop sharing my screen and take it over to Amaya.
All right. Well, thank you, Annie. I'm going to start sharing my screen. Everybody can see it. All right. So the topic that I want to cover today is about grape maturity and fruit sampling. So we are close to hitting Veraison here. I would say by the end of this week, probably we will be Veraison with most of our cultivars. So it's that time of the year to start sampling or to start thinking about sampling for fruit quality. And that's what I want to review and show a little bit of data in terms of Brix, TAs, and pH that we have collected through, I would say, about 8-9 years for these cultivars. And I know that there's very few information published about these values, and I always get questions asked about, you know, what are the targets, where should we be, what can we expect to achieve with these cold climate cultivars. So I'll talk a little bit about that. So let me see, here we go. So the first thing I want to talk about is how to estimate grape maturity. And I would say the most common way is through these three different acids that would be sugar content, acid content, and pH. Those are the most common. There are many others. In my lab, we do a lot of work looking at other biochemical parameters related with fruit quality, such as color or color compounds like anthocyanins, tannins, proteins, phenolic compounds in general. But these ones are the ones that are most useful to determine when would be the best time to harvest the grapes. So the first one is sugar content. We usually measure that in degrees Brix, which is basically the grams of sugars in 100 grams of juice. What we determine with this is mostly glucose and fructose. Those are the two main sugars that we find in grapes. And in general, very general range, we expect that by the time of harvest, most of our varieties are between 18 to 24 Brix when we want to harvest. That's sort of like a very general, broad range of where you want to be. Then we also have acid content. Acid content, we measure it as titratable acidity. So these are the grams of titratable acids in 100 grams of juice that we measure. And the three main acids that we find on grapes, on berries, are tartaric, malic, and citric acid. So those are the ones that we are measuring. We're measuring the concentrations of these acids through the titratable acidity. And then we also have pH. And so pH is basically the negative log concentration of hydrogen protons in the solution. In this case would be the juice that we're testing. And it's also, we can also refer to it as to the active acidity, which is related to TA, but it's not the same as TA. So the pH is correlated with the titratable acidity, but they're not measuring the same things. And I have a note out there that says that the TA is basically measuring the amount of acids that you have, while the pH is measuring the strength of the acid. So you could have same TA and different pH, or same pH and different TA. They tend to behave in the same way. We will see from Veraison all the way until harvest that the acids will drop, and so will the pH, but they are not the same and they are not measuring the same. The pH is really important because it's related with microbial stability. So the higher the pH, the more basic the juice that you have, there's more chances of having microbial sporulation, and you will have, for example, contamination and spoilage of your wine. It has to do with color as well. It affects the color stability of your wines and affects flavor and a lot of other things like protein precipitation. It has, the pH affects a lot of other reactions that are happening, especially when you once already start fermenting that juice. In general, for the white cultivars, we have measured that the pH by the time of harvest is between 3 to 3.4. And for the red cultivars, most of them are between 3.2 and 3.7. Anything over 4 is a little bit worrisome because that's when you have more chances of getting some microbial, higher microbial populations and spoilage of your wine. How do we measure these 3 indicators of grape maturity? Well, you can see, I don't know if you see my pointer, but the first picture on the upper left hand of the screen shows a digital refractometer. So that's how we measure Brix. And right below that on the left lower corner, there's a manual refractometer. This is very common. A lot of growers have this one where you just put a little drop of the juice in there and you just look against the light, the sun, and what the refractometer is going to show you is the concentration of sugars in Brix just by refracting the light that is passing through those sugars. So that's how it works. A lot of you have that. We usually use the electronic one is so much easier. So both of them for for Brix, concentrations of sugars. And then on the right upper corner, I have the picture of one of our titrators that we use in the lab to measure titratable acidity. We use that, but this is a piece of equipment that tends to be a little bit expensive, and unless you have a really big vineyard and you're running a lot of samples and you have the capability of purchasing one of these equipments. The other way that you can measure titratable acidity is by using a pH meter. And so that's the image. I have a small little pH meter there on the lower right corner of the screen. That's a portable pH meter. And so you can actually measure TA by using the pH meter. Again, I already said that pH is not the same as TA, but you can use it and do some calculations. And the way that you would do that, it would be by using a base to basically neutralize all the acid in your juice solution. So the juice that you got from those berries, you would use sodium hydrox, a solution of sodium hydroxide, and then you would measure your pH. And there's a really easy formula. There's tons of videos on YouTube on how to do this. This is a much cheaper way of measuring than having a titrator. So a pH meter is very useful. What is important is that if you have a pH meter, that you have your calibrating solutions and that you take care of calibrating the pH meter every time you use it. The image that I have right in the center of this slide is a new type of titrator. Is an acid titrator. Sorry, it's an acid refractometer. Sorry, this is a new technology that with this device you can measure Brix and TA at the same time. And so this one is specifically for grapes. I found it just Googling it. I knew about it because the blueberry industry uses quite a bit. So this is a great new tool. We haven't used it in the lab, but we are purchasing one right now, and we're going to make the comparisons with a regular refractometer and our titrator to see if the values are similar, but then with just one squeeze of your juice, you can get both readings at the same time. So it looks very promising. We'll be testing it. So that is about how to measure sugar content, acid, and pH. Now, some guidance about fruit sampling. So I just want to cover some very general guidance, like if you're going to be, and you should be, testing your grapes to determine when you're going to harvest, what are the main recommendations? We usually start at Veraison, but I know that it's very time consuming, and unless you're doing this for research, you might want to start a little bit later. So anytime between 2 to 3 weeks after Veraison, you can start. We advise growers to go and sample for testing for sugars, acids, and pH almost every day as you get, within a week of harvest, because things can change pretty fast, especially if the weather is warm. So at the very beginning, you can space those samplings more, but as you're getting closer to harvest, you might want to do those samples more often. Between 100 to 200 berries per block per cultivar is what we recommend. And when I say per block, I refer to the same cultivar, maybe a block that has very similar characteristics. It's very homogeneous. The vines look the same. They're the same age. You know that those vines, sort of behave in the same way. You harvesting at the same time. That would be a block. And so for that, we recommend between 100 to 200 berry samples. If you are collecting clusters, which is the other thing, I would say about 10 clusters would be similar to 100 to 200 berries. When you're collecting the berries, you want to sample from everywhere in the cluster. So you want to, because the berries within the clusters are going to be maturing at different rates, not all the same. So you want to have a good representation. And so from the top of the clusters, the bottom, the back, the shoulders, everywhere, and also from both sides of the trellis, you want to collect berries. So a pretty representative sample of those berries that you have across the panel from different vines and also from both sides of the trellis. We recommend to sample at least 10% of the vines in that block. That's out of all the vines that you have, about 10 of those vines, you would go and collect some berries from them. It's important to collect the samples at the same time of the day. If you collect sometimes in the morning and sometimes in the afternoon, what happens is that in the afternoon, you might have actually higher Brix, so higher sugar, than if you collect them in the morning. We usually try to do it in the morning, and we stick to that time every single sampling that we do. So I would recommend that you just, pick one and then do it always at the same time. Avoid sampling berries from clusters of vines that are in the edges of the blocks, because those vines get more sun and they might be a little bit further along in maturity. So they're not a good representation of the rest of your block. And then the best way to collect the samples is just using a Ziploc bag. You just put your berries in there, you close it. And if you're going to collect a lot and you're going to spend several hours collecting berries, I would recommend that you carry with you a cooler and you keep those samples cool until you are able to analyze them with your refractometer or with your pH, unless you are doing it as you go. Then if you want to store them, I would say do not store them in a refrigerator, but don't store them more than 1 day. So the next slide I have. So here is this slide that I prepared looking at kind of like all data that we've collected for some of the cultivars. I would say probably the most popular cultivars of cold climate grapes. And so what we've done is we've been sampling the same vineyards for, as I said, maybe over 9, 8 years for some cultivars, like the older ones, like Brianna, Marquette, and Frontenac. We have a ton of data, and La Crescent as well. And so what we see in general is when we harvest these cultivars here in Madison, so southern Wisconsin, these are the range of values that we've gotten for about 10 years. So Brianna, this white cultivar, Brianna, tends to have Brix not very high, between 17 to 20. But the TA is very low. It's probably one of the cultivars that achieves the lowest TA. And this sometimes can be a problem because it drops really fast. And when the grapes that you harvest have very low TA, the wine that you make tend to have a little bit more of that Labrusca flavor. And so we're always very cautious to try to avoid getting too low in our TA, because we ended up, having, wines that are probably not as fresh as we would like, or not as fruity and floral as you can get when you have a little bit higher TA and higher concentrations of acids. La Crescent tends to be one that we really struggle with lowering the TA. The opposite, between 13 to 15 is where we usually harvest them. I think that 13 was probably an outlier, a year that I think it was 2012. That was a very warm year and it really dropped. But in general, we have a high, we really have high TA in La Crescent. Itasca, relatively new. Of the years that we've harvested here, these are the values that we get. The TA is a little bit more moderate, between 12 to 13. And then the bricks we achieve between 21 to 22. Marquette, I would say, we usually get into the higher range of bricks. Between 23, 24 is pretty normal, and the TA is a little bit lower than on some of the white varieties, especially La Crescent. Frontenac tends to get higher bricks, around 25. It's very usual that we harvest them between 24 and 25 here in Madison in our research box. But the TA is, again, something that we truly struggle with Frontenac. So anything that we can do, like leaf removal, that would increase the drop of that TA before harvest, we try to implement it every year. And Petite Pearl, very popular this year in Wisconsin. We achieve with Petite Pearl around 20 to 22 bricks most of the years, and the TA is much lower than in Frontenac and Marquette. They are the two red cultivars, and we get between 9 to 11. In terms of pH, it's all of them are kind of close. That's why I say we can see difference in TA, but not necessarily means that the pH changes in the same degree. They're two different things. And so in the case of the pH, anywhere between 3.0 to 3.4 is what we usually get out of those grapes when we harvest them. And just for comparison, I was briefly looking at the growing degree days that we get here in Madison for those of you that are somewhere else. So I have 5 years that I was looking at for growing degree days accumulation from April 1st to September 30. And we usually get about 2,600. I said one of the years was 2012 that we got the most. It was almost 3,000 growing degree days from, again, April 1st to September 30. But in general, I would say between 2,600 and 2,700 is where we get here. So those are the values that we get. If you have lower growing degree days accumulation, you will probably have lower bricks accumulation. And with that, that's everything I have. I'm going to stop sharing, and I think that we're going to answer. I don't know if Annie wants us to answer questions now, or maybe we'll leave all the questions to the end.
We're going to save most of the questions for the end, but the one question that came up during your presentation was from Craig. He asked, how much is the COM-BP meter? Am I reading that correctly?
Let me see. I don't know if I see it.
It's in the chat.
I was looking at the acid refractometer. I probably used that one. I was looking online, and I think I saw somewhere between $250 and $300. But as I say again, I haven't used it. I know that the blueberry industry uses it quite a bit, and when I checked on their website, and they're probably in multiple brands. I didn't spend a lot of time looking at all the brands, but I would say that I would still like to validate it. I haven't used it, but in their website they had it specifically for grapes, so they're using it at grapes as well. I wonder whether maybe it's something that in California or in Washington they're using more commonly. But it seems like a great tool if you can get your TA and your bricks at the same time with only one button.
Thanks. Okay, so we'll just go into the next presentation, and then if you had more questions, if you think of questions later for Amaya, just type them into the chat box because we will get to those at the end. So I wanted to review some of the bird management options that we have because I do get a lot of questions about this time of the season, even though admittedly it's a little bit late to be selecting your bird management methods, especially if you need to order some. But this is definitely a topic of conversation this time of year. So I have been spending some time reading the literature to see what studies have been done that actually compare how effective these bird management methods are. And it's really interesting. A lot of the research has been done in California and Australia. There's not a lot of research on bird management methods in the Midwest, which I did find interesting. Maybe there's just 1 or 2 research labs in California that have this as a focus. I don't know. But I did have to kind of dig far back in the history of viticulture research because some of these studies are pretty old, so they date back to the 70s. I learned a new vocab word. It's called frugivorous. Frugivorous, I think I'm pronouncing that correctly. And so that just means any animal that feeds on fruit. Herbivores can be frugivorous as well. So humans are frugivorous if we eat fruit. All right, so we're talking about frugivorous birds that like to eat grapes. A lot of these birds also like to eat blueberries. So a lot of the studies I found were comparing grapes and blueberries and sometimes cherries as well. Anyway, so there are a number of methods available. They do differ in effectiveness, and for some of these, there's not a lot of research showing how effective they are. The main one, of course, is bird netting. The second one is methylanthranilate as a foliar spray, and this is basically an artificial flavoring chemical that can be sprayed. It is labeled for application to fruit crops, so it can be sprayed on grapes. And the idea is that it can deter birds, but we'll see if it actually works. Artificial sounds such as cannons and gunshots, I know that those are popular in Minnesota. Another one that's popular up here is distress call mimicking sounds. The popular piece of equipment that I see a lot is called bird guard. Another is lasers, and then there's drones, and of course the traditional balloons and kites, which they don't seem to work very well. At least I'm not on my family's vineyard at all. And then the introduction of predatory birds such as owls, and in other parts of the world, falcons. Keep in mind there are a lot of different factors that impact bird pressure, and this paper that I cited here from 1972 goes into some of those factors that can affect how many birds we're actually seeing on the vineyard, how much pressure is there from the bird population. So it depends on species, the environment, the time of day, the grape variety, the sugar levels of the grapes, the foliage. That was an interesting one. The amount of foliage in the vineyard can actually affect how some species of birds get into the grapes and how some don't. Surrounding vegetation, like is it surrounded by cornfields, swamp, woods, whatever, and then weather. So is it hot? Has it been really dry? If it's been really dry, maybe the birds are going to gravitate towards the vineyard because there's not a lot of other food sources. If it's been wet, maybe they're going to be elsewhere. So a lot of different factors to consider. This paper was very interesting. It did a comparative study of 3 different methods. This is from 2007. So they were comparing bird netting, distress calls like bird guard, for instance, or visual deterrence. And they found that with bird netting, the vineyard only experienced 2.3% damage, so that's very low. In other words, that would be like 97.7% effective. When they combined the distress calls with the visual deterrents like balloons and kites, they got 5.7% damage. And then with the visual deterrents alone, without the distress calls, 13% damage to the vineyard. So what that's showing is if you're going to be using the visual deterrents, it's going to work a lot better if you add the distress calls into that system. But in this study, bird netting still did the best job. One of the reasons for this is not all of the birds responded how we want them to respond to the distress calls. Some of the bird species that they were looking at had no effect of the distress calls, and some of them were actually attracted to the distress calls. Robins, for instance, they actually tended to come to the vineyard when they heard those calls, rather than scattering. Bird netting is not discriminatory. Bird netting is going to exclude basically any bird in the vineyard, regardless of species. So this is a picture of me and my husband using the netter getter. Full disclosure, my dad and uncle actually invented the netter getter, and my uncle is Paul Claude, the one who manufactures that in Iowa. This is not an advertisement for the netter getter. However, I'm just showing it as an example. A lot of vineyards in Minnesota do have net machines, whether it's it's this product or one that they have made themselves, it makes applying net to a vineyard a lot more efficient. So I'm not going to go into detail on netting. I think at this point we all know what netting is and how to use that. But bird distress calls, so one example is bird guard. And there's a lot of questions about whether or not these are going to be as effective as netting. But there have actually been quite a few studies on bird distress calls and how effective they are. And these studies have found that they're going to be effective for about 2 to 6 weeks before the birds habituate to them, meaning the birds just start to get used to them and think, oh, okay, well, I keep hearing these sounds, but then nothing bad happens. So they'll keep going back to the vineyard. So then one of the literature reviews on this said that some of the studies on distress calls found these effective, but about half of them found them ineffective. So there's a lot of variation in how effective this is. Maybe the vineyard down the street from you has very good luck with bird guard, but then if you try it, maybe you don't have the same results as they do. So it's just important to realize that this method varies a lot from vineyard to vineyard. It's been found that they have better performance when integrated with other methods, such as cannons or such as the visual aids. And they found that robins do not always disperse. They might actually congregate, like I mentioned before, and they tended to be more effective on starlings. That's from a study from 2007. So in summary, this tactic may or may not work well on your vineyard, and there's probably a lot of different factors at play that are impacting the success of these in all of those different studies. Chemical repellents. I had a conversation with somebody about these just the other day. So the active ingredient in these is methyl ethranolate, and an example of a product that contains this is avian control. So there were 4 studies that I was able to find on this, as well as a literature review that basically said, yes, these are the only 4 studies on this product that they could find. 3 of them found it ineffective, and 1 found it to have some effect on certain species, but certainly not overall effective on all bird species that might eat our grapes. And so right now, I cannot recommend this product because 3 of the 4 studies that I've been able to find found it ineffective. There is one vineyard in Minnesota that uses it, and they feel that it is effective on their vineyard. I won't dispute that. Just think about, if you try this method and you find it effective, that's great. Think about what other factors might be impacting that as well. So here's some things that you could read about the effectiveness of those chemical repellents. Another one that's coming up more recently in the research is using drones to scare away birds from the vineyard. So this study from 2019 was certainly very interesting. So they flew drones, also called unmanned aerial vehicles, UAVs, over the vineyards, and they attached speakers to them to actually play the distress calls. So the drones are playing the distress calls, and they attached taxidermy crows underneath each UAV. And they found that one drone can effectively cover 61 acres. So if a vineyard is larger than that, they're going to need a second drone. They also found that one UAV flight could deter silver eyes, which is one of the species they were studying, for 15 minutes. And so that would mean that if you're using a drone, you would need to be out there flying it every 15 minutes. I'm not as up on UAV regulations as I used to be, but the last I knew, you still need to actually be there watching the drone while it's flying. It can't just be flying on its own. That may have changed, but the last I knew that was the regulation. So you would have to be out there quite a bit. This study was also done in Australia, so it's not our birds and not our environment. But I do find this to be an interesting field of study. Another one is lasers. And so I would say that, in my opinion, this is a promising area, and it's very interesting as well. It's not commonly adopted yet, and I couldn't find much of any research on it, to be honest. Basically, what I found online was a couple of anecdotal stories of people saying that it worked for them on their farms, not in vineyards, but in blueberry farms. There was one study that had a paywall, so I couldn't access it. So I linked one article down here that maybe you can read it and find it interesting. I know that there is one vineyard in Minnesota that used lasers in 2019, and as far as I know, they are planning on using it again in 2020. So I will look forward to hearing what results they get from that. I know that they had to install a couple of different lasers because of the size of their vineyard. And I just feel that this is a promising area, but there does need to be more adoption of lasers on vineyards before the effectiveness of this technique can really be evaluated. So just keep your eye out on research and stories about lasers on vineyards. Just a few notes, if you're going to be testing new bird methods on your vineyard, let's say you're sick of using netting and the time it takes to take it on and off, maybe you want to try some other things. Just keep in mind, as far as we know, based on the research, netting is still the most consistently effective and reliable method because, as I was saying before, this is a physical exclusion. So it's going to exclude basically any birds that are in the vineyard as long as they're not hopping up from the ground into the net. Whereas other methods, they're really dependent on the species. Each species reacts differently to these methods. So right now, you know, if you really want that complete effectiveness, netting is still the way to go. Keep in mind, bird pressure varies each year. So if you try a new method, just be cautious not to be too quick to attribute low bird damage to a new method if you don't have an untreated area of the vineyard to compare it to. So let's say if you apply avian control, that spray product to your whole vineyard, and you don't have a lot of bird damage that year, maybe it's due to that new treatment that you tried, but maybe we just didn't have a lot of bird pressure that year. So before you become too attached to a method and too reliant on it, just make sure you understand all the different things that could be impacting the success of it. So I would give a new method a couple of years in your vineyard before drawing too many conclusions about its effectiveness. So anyway, that's my take on bird management methods right now. Hopefully a couple of those things, like maybe the lasers or the drones, could be a promising method in the future once we get more exposure to it.
Well, thank you, Annie. And I guess I'll start with mine. Or do you want to take questions now?
There aren't any questions.
Okay. So let's see. Okay, there we go. Okay, well, all right, everybody. Thank you for joining us. And I'm Christelle Guedot. I'm the food crop entomologist at UW-Madison. And I'll be talking about the insects that you should be paying attention at harvest. So you might have other insects that I'm not covering here, but here are 3 that we are paying close attention to in general at harvest for grapes. So I'll start with the multicolored Asian lady beetle. And I just want to show you this slide because it shows you the different color patterns that they have. There's really a lot. So trying to recognize them based on their coloration or the number of spots is not going to be the way to go. If you can see my mouse moving here, it's this kind of M on the thorax of the ladybug that you can help you identify that it is multicolored Asian lady beetle. But that's about it. And as you should know, hopefully, all lady beetles are known to be biocontrol agents. So in general, that's why they're here. And multicolored Asian lady beetle was introduced for biological control to begin with. So that's what we usually see them for. But then we also have the problem with this species in particular that wasn't intended, is that they aggregate in the fall or early fall, late summer, on those late season fruits. And the reason why is because they really need sugar before they go overwinter. And as you know, they overwinter in your houses. So there's been research showing that they need this sugar solutions to survive for the winter longer. So we'll find them in grapes, in apples, and raspberries. Where, from a standpoint of grape, there's a study. So most of the studies I'll talk about today, I think maybe even all of them, are from Minnesota, actually. But this paper by Galvin et al. In 2008, they put intact Frontenac grapes. They exposed them to one lady beetle for 24 hours, and there was no evidence of damage. And I'll show you a slide about that after. And then another problem with those lady beetles that most of you know is when you're making wine and you're crushing those lady beetles, we have what we call ladybug taint. And I'll talk about that in the following slides too. So this was a study another one where they looked at, First of all, the studies have shown pretty much that they don't cause the initial damage, that it's pretty much using the damage that's been caused by birds or other insects, and then those beetles come in and feed on those wounded grapes. And then this other study that showed that they will settle on damaged grapes way more than they do settle on undamaged grapes. And so you can kind of see that here in the red rectangle. This is on damaged grape. You have way more that do settle on those than on undamaged grapes. So confirming yet again in another way that they go where the damage is already there. So when we talk about that ladybug taint, we all know that it's a negative impact on wine quality, and it's caused by the hemolymph. The hemolymph is the blood of insects, and it's this compound, 2-isopropyl-3-methoxypyrazine, that is found in the hemolymph of those multicolored Asian lady beetle as a deterrent for predators. And so that's what gives them the wine that taste, that bitter taste. There has been some thresholds for red wines. It's 1.3 multicolored Asian lady beetle per kilogram of grapes. For whites, 1.5 per kilogram of grapes. And what a study has found is that almost 2 lady beetles per kilogram of grapes was detected by 10% of the human population. And that corresponds to about a third of a beetle per Frontenac cluster. So there has been remedies that have been suggested in research. For example, activated charcoal in white wines, deodorized oak in red wines, oak chips in both red and white wines. I don't know if anyone has implemented that, if people have had successes or failures with this. I don't know, it's not very clear that it works really well, and I don't know if people are ready to implement those for remedying this taint. So if anybody, even in the panelists, have suggestions on that at the end, we can talk about that. So again, they come where there's clusters that are damaged and when there's sugar for them to get. So they correlate with the increase in percent cluster with damaged berries. And they arrive 2 to 3 weeks before harvest. So if you look at this graph, that's the, in black, that's the lady beetles on sticky cards. This is the cluster with damaged berries. And this is the lady beetles in clusters. So as you can see, everything is correlated, and it goes, it increases as the number of clusters with damaged berries increase. So for monitoring them, you have those yellow sticky cards. There's also these pyramid traps that you can see down here. And from the sticky cards, there's been a study that showed that the yellow is the most attractive for those lady beetles, similar to the green. So, but because the yellow is a very common one, that's the one that we recommend. So what is suggested by this paper from Galvan et al. In 2007 is that you want to increase the sampling frequency in the last week before harvest. The actual time for sampling during that last week should be based on the pHI of the insecticide you're going to use, because obviously one insecticide with very short pHIs because we are at harvest. So for example, if you have a 3-day pHI insecticide, you want to sample actively 5 to 6 days before harvest to provide you time to implement the control. And the action threshold that are suggested here are 0.05 to 0.6 multicolored Asian lady beetle per cluster. If you get that, then that's when you should implement an insecticide spray. Sanitation is also something that you can do. You can check the cluster to dislodge them. You should cover your bins where the cluster are held as much as possible because they will go into those bins. You might float the clusters in buckets of water to dislodge them, or you can vacuum the cluster to remove the beetle. So these are different strategies that people have used. There's this survey, so there's a link down there if you want to access it, but in Minnesota, what was shown is that the growers that use pre-harvest IPM strategy, whether it was sampling and a single spray for multicolored Asian lady beetle, in grapes may save at least $200 per acre when compared with growers that rely only on physical or mechanical removal of beetles. So something that you should keep in mind and look into this survey if you want to know more about the different strategies, but that's an interesting comparison of the costs. I will not talk right now about the insecticides that are available, because what I did is at the end, I have a table with insecticide that kind of go over the 3 insects for what I could find. So you'll have that at the end. So I'll talk quickly about managing social wasps in Vineyard. If you have social wasps, you probably have paid attention to what they look like. And what you will find is that you have paper wasps, which are the ones that are up here with the dangling legs. And they are Polistes, that's the genus of those wasps. Yellow jackets are the more robust wasp that you see here. That's the Vespula. We have bold-faced hornet, that's Dolichovespula, and then the true hornets, Vespa, that are actually, we don't have a lot of them here, and this one is introduced. So the problem with the wasps is that there are reports of losses due to wasps, whether it's in grapes or other fruit crops, that can go up to 100% in grapes in some older reports from 2011. They can vector sour rot in grapes and human diseases like salmonella. So there's been studies showing that they are vector of those pathogens into the grapes. Of course, then there's for pickers and people that are visiting, touring around the vineyards, anaphylaxis threats from the, what do you call that? I forgot the term, but you know what I mean. Allergic reaction. And then also they can be very detrimental. In Wisconsin, we have reports that some years they can be the most detrimental pest in vineyards, but it varies from year to year. And then the management options are very limited and do not provide adequate control. And those management practices are really all listed here. So number 1, there is no labeled pesticide for wasps, because technically wasps are beneficial insects. They feed on pest insects. They are not doing any kind of harm to anything. Only at this time when they come and feed on our grapes can they be a problem and as a nuisance problem. So they're not considered a pest. So there's not any pesticide that will have wasps on the label. And the best option that people will tell you is to locate and remove the nest early in the season. So that's something that's not easy to do because it's not easy to find the nests. You want to minimize the injury from other pests because they can come after the grapes are already damaged. And we'll talk about that in a little bit. Prompt picking is an important one. Removing overripe and damaged fruit. You'll see a lot of wasps that come to dropped fruit and damaged fruit on the ground. There are some insecticide you can spray, but that's what you would spray on the nests, right? So when I mean there's no labeled pesticide, that would be for grapes. If you're looking at grapes pesticides, none of them have wasps on their label. You can buy those kind of insecticide that you spray on the nest to kill a wasp nest. And then you can power wash with water, you will knock them down anytime you see those kind of nests on your houses, on eaves or places like that. And then there's trapping that people have done with on and off success and different baits and different traps and things like that. So what I will talk to you about now is the research we've been doing in Wisconsin regarding those wasps in grapes. And so what we started with really was trying to figure out what wasps we have, because as I mentioned, we could have paper wasps or yellow jackets or hornets. So there's different types that we may have. So what we did is we sampled and we looked at what we find in grapes. And I want to bring your attention primarily to the different pie charts represent different years. We have 2015 up here, down in the middle, 2017 and 2018 up on the right. And really you can see that the proportion of those changes from year to year. So there's some that are more predominant, like this Vespula maculifrons, but you have also others that in 2015, this Vespula vidua was very predominant and we don't see it anymore. So there's a lot of variation, and that goes back to what Annie was saying about the birds, where whatever you implement might not be really something that's due to the fact that you implemented a strategy, but just the natural variation in populations and which ones are more prolific one year from the other. And that's well documented that it changes, and some are on biennial cycles and things like that. But the main species that we have is about 5 species, and I'll go back, but this Vespula Vespula germanica, the German yellow jacket. We have maculifrons. We have Dolichovespula. That will be our bold-faced hornet. And we have others that then change a little bit here and there. When we look at all of our wasps together, this is kind of the seasonal phenology we saw in 2015. And as you can imagine, the populations take a little while to build up. At around Veraison is when they start ramping up. And then really, they keep going. If we had kept going, which we did in years after, they keep going up and up and up into September and even October. So they become a problem because they overlap with the harvest at almost their peak. So we conducted assays to determine whether those wasp species, the more predominant ones that we saw in Wisconsin grapes, were actually able to damage the grapes or not. Because they're always thought about as insects that come after the damage is already done, like the multicolored Asian lady beetle. But it was a little bit doubtful to me that that was the case. So we did some lab bioassays where we put wasps on grapes. We had 4 cultivars, Somerset, St. Pepin, La Crescent, and Marquette, to cover our wines and table grapes in red and white. And we just looked at damage versus intact grapes, not together, separately, to see if there was a feeding on damaged or intact grapes. And so what we have here, this is the bold-faced hornet. And what you have down here is on the y-axis is the average % of damaged grapes. And then the first panel is the damaged grapes. The middle panel is the intact grapes. And then the third panel here is seeing if those that were exposed to damaged grapes and then exposed after that to intact grapes. And here we're only looking at the damage they do on intact grapes after they have been exposed to the damaged grapes. And that's what we call the learning. And so the gray bars show you no damage. The pinkish bars show you only surface damage. And then the red bars is internal damage, where they really were digging into those grapes to feed on them. So here with the bold-faced hornet, what we see is that there is significant damage on the damaged grapes. There is no damage at all on those intact grapes in the lab. But these are the ones that we documented that learned. So here they're exposed to damaged grapes, intact grapes after they've been exposed to the damage, and now they are able to damage those grapes. And something like Somerset seems to be, or even Marquette, seems to be particularly vulnerable to that learning, where they're able to now damage those grapes that are intact when they were not able to do it in the first place. When we look at Vespula germanica, the German yellow jacket, or Vespula maculifrons, I don't present the data for maculifrons because it's similar to germanica. But here in this case, again, they feed extensively and do a lot of internal feeding on the damaged grapes, but they also do feed damage onto the intact grapes to some extent, very deep feeding, and also on all of the cultivars we tested. And then in this case, we did the learning, but really we're not surprised because they can do damage the intact grapes. So not so much here to get out of this. We also looked at what we call a push-pull strategy. So what we were interested in was looking at repellents and attractants. And I'll go over this quickly, but we're trying to repel them from the grapes and attract them in traps. We had different designs here. We had control, they're just repellent, attractant on the perimeter, and then attractant and repellent pulled together. And what we found here is that in our monitoring traps, this is the control here. And so anything that's similar to the control with the B letters would be that there's no effect. And that was the repellent alone. And what we found is that the attractant decreased the number of wasp per traps in those monitoring traps, similar to the push-pull, which means that considering that control and repellent were not significantly different, and attractant plus repellent were not different, that we really have more of a mass trapping effect that happened here, where the repellent didn't really have an effect, which was quite surprising to us, but interesting to see. Down here, it's showing you that we pull a lot of wasps in those traps, in those attractant traps in the mass trapping. We also tested the repellent by itself, and what we actually found when we just put repellent in a monitoring trap And that repellent, actually, I didn't mention what it is, but it's that methylantranolate. It's the same than what's in that bird repellent. And what we found is that when we use the repellent, we have a reduction that's not significant here when we look at all the wasp species. But when we look at species separately here, what we see is that for the bold-faced hornet, we had a significant reduction with the repellent, and that's what you have here. So all combined, we don't see a difference. But when we pull out the species, we start seeing differences, just like Annie was saying with the birds, that for some species, bold-faced hornet, we have an effect of the repellent, but not with the other species. Okay, I'll finish quickly, and I'm sorry we're running late on time, but just managing spotted-wing drosophila in grapes. So I just want to reiterate for the Wisconsin people, because you've seen the research we've done several years ago. But we tested on our cold climate grapes, and you have the cultivars down here: Frontenac, Marshall Foch, Marquette, La Crescent, St. Croix, and Edelweiss. We looked at whether we have adults in the vineyard. So we put traps in those different blocks of different cultivars. And we did find spotted wing, I was going to say wasps, everywhere. In all of those cultivars, we do find adults. We also find larvae, but in a very, very, very small amount. So we have very few larvae. There's no difference between the cultivars, but that would be about 2, at the most about 3 larvae per kilogram of grapes. So very few larvae. So then we went into the lab and we looked at similar, not all of them, unfortunately, but we looked at similar varieties. And here we looked at undamaged, same thing what we did with the wasps. Undamaged grapes, damaged grapes, we put spotted wing on them, and we compared those different cultivars, but looked also with raspberries, and we looked at whether or not we had eggs, larvae, and adults. I'm only going to show you the larvae, but it's the same for all of them. What happened with all of those cultivars is that we have spotted wing Drosophila larvae and adults and eggs in damaged grapes, and we have almost none in the undamaged. We had, I think, a total of 2 flies that emerged, and we think it's because we had a crack in one of those grapes. So really, we have no impact on the undamaged cultivars that we have here. There's a study that, the study that came out from Minnesota with Bill Hutchison and Dominique Ebbenga, which you probably have heard her study. But there's a similar kind of trend where most of those cultivars, and they tested other cultivars, we're not seeing a lot of larvae or very few larvae in the field. So there's really a tendency overall that our grapes are not susceptible to spotted wing here. So what I would say is that once the grapes are damaged, it's fair game to those flies. So if you have splitting, cultivars that are prone to splitting or heavy rain and you start having splitting, or if you even have a crack at the pedicel, that's an entry wound for those flies to come in. So the recommendation that we have really, from what I showed you, we have flies everywhere. So monitoring for flies is really not the option. Flies are everywhere, and we're not even trying. Once you have the first fly, there's no need to monitor anymore. The flies are there. There's nothing major to do about that. It's sampling for larvae within the fruit. So if you have larvae within your fruit and you're very close to harvest and you have those Brix values that Amaya was mentioning, you might consider harvesting as fast as you can. So of course you have the balancing act, and Amaya and I were talking about that yesterday, of when would you do that, but you have to think about that. If your fruit is compromised, is splitting, you're gonna have, because those flies are everywhere, you're gonna have larvae in your fruit. So our recommendation is to apply an insecticide only if your fruit is infested, not if you have flies in traps. And that goes back to the webinar we had on July 15th with Megan Hall about the bunch rot management, where she was recommending to monitor. So you're going to have, again, a balancing act to do here between the adults and the potential for bunch rot that happens in the transmission of the pathogen, and just having larvae in the fruit. So I just want you to think about that, because these are our recommendations from a standpoint of larval infestation, but it's not what Megan Hall would suggest from a bunch rot management standpoint. So again, a balancing act for you. This is just to show you what you would do to sample the fruit for larvae. So you put them in water, crush them in a plastic bag, wait an hour and see the larvae that float inside. I just wanted to show you this because it would be 4 cups of water to a 1/4 cup salt. So that's something that you can refer to if you're doing this fruit sampling. We call this the salt test. And then I'll just end with this slide. And this is showing you a subset, of course, of chemistries that you can use. I started off with what we have for multicolored Asian lady beetle. Again, here I'm providing you with insecticides that have different mode of actions, but also short pre-harvest intervals, at least as short as possible. And then the efficacy for those 3 different insects that I mentioned. So in orange, you have multicolored Asian lady beetle. For wasps, unfortunately, I don't have a lot because nobody tests on wasps. So there might be a little more studies that are coming out of Michigan now, but I haven't seen anything yet on that. So I didn't find a lot. So when I say no data, that doesn't mean it's not at all there. It's just that I haven't found anything yet. And then you have for spotted wing in purple. So I hope this helps in making decisions if you have to deal with those different insects. And I'll end with that.
All right, awesome. This is such good information. So we are almost out of time, but we have a few questions, and feel free to keep entering your questions. So I'll ask this one first for Amaya. When is the best time to send in tissue samples for analysis of nutrients?
There's 2 times where you can collect tissue analysis for nutrient content. So the first one is at bloom. And we talked about this on previous webinars, I think, and it's also part of, in a lot of the articles that we wrote on our newsletter and our website, is you will collect the leaf that is opposite to the first cluster. So the cluster that is closest to the corner or to the base of the shoot. That's a bloom. The second time would be now during veraison, and then you want to take the first fully expanded leaf from the tip. So if you select one shoot, you count the leaves back. It's about number 7. It was probably the most recently fully expanded, and that's the leaf that you want to collect. And you want to send the petioles. So you collect the blade and the petiole, and then you separate them as close as possible in the union between the petiole and the blade. And you collect all of that. It's about, we recommend to send between 150 to 200 petioles per sample. But you can watch that video that Anna just said, and there's also some information on our website. I can put it on the chat box.
And you said 150 to 200 petioles per sample, and the U of M tissue lab says like 50 to 80. So I don't know why there's that lack of consistency there, but
I think it's mostly about getting a big sample for representation. Mm-hmm. And that's the reason why we always recommend to send more.
Okay. All right. And then any advice about deterring turkeys?
Not from me.
Not from me either.
Yeah, I don't necessarily either, except that I am working with somebody who's having issues with squirrels in their grapes, and the best thing that I've been able to recommend to them, short of like installing a fence or an electric fence around the vineyard, which that is an option for you, trying to secure that netting underneath the canopy so that it's harder for the grass to get up in there.
How about getting a dog?
Yeah, getting a dog too will help. So maybe those are helpful. Somebody asked, what does SWD stand for? That stands for spotted wing drosophila. So that's that invasive fruit fly. She also says, I have raccoons and I have an electric fence planned. And I know somebody who has actually a few growers who have electric fences, and they can be fairly economical. They don't have to be big and fancy. The people I know who have them say that they're using them to deter deer. So the deer might not be able to necessarily see them, but they will feel that electric fence when they walk up to it. So maybe it would work for turkeys too.
I also see a question from Craig about how about netting for insects. And I would say that yes, netting is always something that you can do. The physical exclusion that Annie mentioned for birds, it's all about the mesh size, right? So depending on what it is that you're trying to exclude, it's the mesh size, and it's also the big part of that is that you want to apply that before you have a population. So especially with insects, you don't want to be putting your netting in September and expect that you're not going to net with that any spotted wing drosophila, right? So it depends what it is that you're trying to exclude. You have to make sure that there's nothing, because if you trap them inside, then they go to town. So that's the part that's really important to think about, and the mesh size. For spotted wing, for example, it's 1 millimeter mesh size that you need to apply. For other insects like wasps, obviously it could be a much bigger mesh size. So it depends on the size of your insect.
Another question, do you recommend hanging traps for wasps and SWD?
So I don't know if the question means hanging traps for, I'm assuming for mass trapping. Is that correct, Teresa? I'll answer to that, and then if it's for monitoring, we can talk about that. But for mass trapping, I would say that for wasps, that's what we applied for a Department of Ag grant to assess that. Because from what we have seen, okay, yeah, the question is for mass trapping. What we have seen with our push-pull strategy is that the mass trapping actually decreased the population of wasps in our vineyard. For spotted wing, there's been several research projects that looked at mass trapping. Unfortunately, what we have, and it's not for grapes, it was in other crops, in blueberry and raspberries, I think. And the problem we have is we don't have attractants that really are better than the fruit. And so what you tend to do is attract them to the area, but you still have damage next to it, and you still have, you have competition between your trap and the fruit. So you still will have plenty of damage in your vineyard. I wouldn't recommend it at all in vineyards, because like I said, our recommendation really here is that the varieties that we have are not affected by spotted wing unless they are damaged already. So our recommendation is to prevent damage as much as possible and try to go that route as opposed to mass trapping. But the research is not conclusive for spotted wing. I think that's our last question, huh?
Looks like it. If you have additional questions that come up after we log off in a second, feel free to email any of us with your questions. Thanks also to the Wisconsin Grape Growers Association for helping get the word out about these webinars and helping us plan them initially, specifically Anna for her help with that. I guess this is our last one for the season, so I wish everybody luck with a great harvest this year.
Yeah, and thank you, Annie, so much for moderating all those sessions and coordinating and organizing. It's been a pleasure. So
yeah,
thank you, everybody,
for you guys for speaking and your expertise. I think it's been really valuable to combine all this knowledge from 2 different states.
I agree.
Yeah, it's been great. Thank you for everybody that participated.
Yeah. Thank you, everyone.
All right. Bye.
Bye-bye.
Bye.